Now, what is genetic engineering? Genetic engineering is the process of manually adding DNA to an organism. Now, why we transfer DNA from one source to another source? इसकी क्या वजह है? The main reason is that actually we are going to introduce one or two novel traits that are not present in the target organism. Now, if we study the history of genetic engineering or gene manipulation, we will find that even hundreds of years ago, humans had tried to indirectly modify the DNA of the organisms. And it is evident by the domestication of different type of animals and the production of good quality of seeds to enhance the growth and yield of agronomic crops. So this modern subject of gene manipulation, it was started around mid-70s when scientists were able to directly transfer gene from one source to the next source. So now this term genetic engineering in literature, some authors consider that this term it is a bit emotive. It means that dealing with to arouse intense feelings and fascinating one, and sometimes they consider it as a trivial, that is having little importance. So because of these reasons, no different authors they have used other terminology like gene manipulation, gene cloning, recombinant DNA technology, and even genetic modification. But overall, it deals with the isolation, manipulation, and expression of the target gene into a suitable host organism. Now, in many countries of the world, there is a precise legal definition of gene manipulation, especially in the Western countries. For instance, in UK, ये जो जीन मैनिपुलेशन है इसको डिफाइन किया गया है कि ये एक ऐसा प्रोसेस होता है जिसमें हम ऐसे कॉम्बिनेशंस बनाते हैं जो कि हेरिटेबल होते हैं. हेरिटेबल मीन्स दैट दे कैन ट्रांसफर फ्रॉम वन जनरेशन to the next generation, and these heritable combinations they are produced by any means, especially outside the cell, by using any bacterial vector, phage vector, or any other system. And when it is incorporated into the host organism, then it is capable to replicate there. So we can define gene manipulation as the process of new combinations of heritable material by the insertion of nucleic acid molecules produced by whatever means outside the cell into any virus, bacterial plasmid, or the vector system, so as to allow their incorporation into a host organism in which they are capable of continued propagation. Now, what are the different applications of the genetic engineering? Although we will discuss this topic in more detail at the end, but here I will briefly explain that this genetic engineering, it is having applications in both basic as well as in applied research. For instance, in case of basic research, in the laboratory, it is used to study the mechanism of gene replication and expression by using different type of host organism, either prokaryotes that are mainly bacteria, and eukaryotic microbes like the yeast, and even one can use animals and plants as a host system, and even their viruses. Now, in applied research, genetic engineering it is used to develop microbial cultures that has your gene of interest and that is capable of producing different valuable end products. Now, what type of end products? For example, very common human insulin, human growth hormones, variety of vaccines, and even the enzymes that has industrial applications. At the same time, genetic engineering, it is also very valuable to produce transgenic plants and animals so that you can introduce desirable traits so that the agriculture sector will provide some good crops, good animals to the people. And the most important use nowadays, it is in environmental biotechnology. For example, we can introduce genetically modified microbes under controlled conditions so that they can degrade those pollutants that are toxic to the living organism. So microbes, they can degrade such compounds. Why? Because we have introduced those genes that can target those pollutants. And then most importantly, that we can also cure different type of genetic disorder maybe in near future by the use of gene therapy.